And so our next talk moves on to a completely different form of life, scales of artificial life and their possibly signatures by Lana Sinpayen at the Sony Computer Science Laboratories. Lana.
Thank you. So I'm going to talk about my scientific field, which is called artificial life, and how I think we can use it to define some new forms of biosignatures. So first, a brief self-introduction. I'm a researcher at Sony Computer Science Labs, and I'm also an associate professor at Japan's National Institute for Basic Biology. I used to do robotics, then I moved to computational neuroscience, then to A-Life, and now I'm doing mostly astrobiology. So if you know me by name, it's either because you know I'm doing A-Life research, or it's because of my talking dog. But today, I'm going to talk about A-Life. So as you have probably guessed from the talk that we had before, we don't have in the life sciences a consensus about the definition of life. What we typically do is we have a list of attributes that we tend to think of as signs of life, and we take some of these attributes, and if a thing exhibits these attributes, we would say maybe it's a life. In artificial life, what we try to do is to try to build life, to try to understand what is life. Unfortunately, we have even less of a consensus about what life means. So that leads to two issues. The first is, how can we build something when we don't even have a definition? The second issue is, we're trying to build life. How do we know if we have succeeded if we don't even have a way to evaluate what we are building? And the answer to both of these questions is, I know it when I see it. And I think that's true for astrobiology as well. You can have the best, most exciting measurements about an exoplanet. As long as people cannot see it, your measurements will be controversial. So what is artificial life? We have three main fields. The first is called hard A-Life, so it's everything that is robotics. Second is wet A-Life, so chemistry, physics, biology. For example, here you have some droplets that are moving around and doing this so-called herding behavior, but this is just chemistry, there is no life here. And then we have software, which is where I am. So everything I'm going to talk about today is happening in computers. And then the little plus one here is art. So when you ask questions like, what is life? You cannot avoid cross-contamination between science and the arts. So I'm going to talk about an in silico approach to how to detect life. And what I want to say that even if we don't have a definition, you can work from this list of attributes and try to define measures that tell you whether this attribute is present in an environment or not. One attribute at a time or a combination of the attributes. First, I want to talk about the project that made me change my mind about how I think about life. The most recent project that made me rethink my approach to what is life. So I use something called the Neural Cellular Automaton, NCA. Very briefly, it's an AI that tries to learn some laws of physics for a simulated world. So you tell it, this is my initial state, this is my final state, and find some laws of physics that will bring me from initial to final state. So what I did is I asked it to learn some laws that can take me from this egg, which has some information in it, get to make this image of a fish. So this is just some 2D patterns. It's not really supposed to simulate any kind of real life. And then from this fish, get me to a fish that has laid an egg. So basically I asked it, find some rules for self-replication. Then I can isolate this egg, and you isolate it, and from this egg you should get, again, another fish. So initially I was very happy because, will this play? No, it's not playing. Oh no. So this was a video of the self-replication. And I have some other videos, and I suppose they won't play either. That's very sad. So anyway, initially I was happy because the self-replication worked. You could have some first generation, you take this egg, you develop it, it becomes another fish. You can have as many generations as you want. And what made me happy is that I could see some mutations happening. I didn't ask the system to learn any kind of mutations. You can make the system completely deterministic, no randomness, and you still have these mutations happening. For example, here there is initially the fish is supposed to have 3 stripes. When you look here, you can see that it's almost reduced to 2 stripes. Here it's almost 4, etc. But then I became a bit disappointed in the system because when you have mutations, you think about evolution, and I wanted this to evolve into something interesting. And I found out that this system only produces fish. Doesn't matter what you do, whatever you put inside. So you can try putting some different type of patterns. They just become fish, and then you're stuck in this fish attractor. And when we think about life, we think about the opposite of that, right? When you have, for example, you have this river, everything goes downstream. The thing that goes upstream, you want to call that life. The thing that avoids the attractor where everyone goes, that's life. So in this fish world, everything that is not a fish would be life. So I went back to simpler simulations. This is a simulation of an artificial chemistry. So you have some atoms, ABC, some molecules, AB and ABC. You define the rules that define how these chemical components interact with each other. And you let the system settle and you get to this equilibrium. You measure the concentrations of these different chemicals. And so this is the equilibrium of this simulation. And the question is, can you get the system to a point where it's not in equilibrium? Can you avoid this powerful attractor? So in this system, I put some protocells. Here, a protocell is defined simply by having an inside and an outside. So there is a membrane. The membrane is made of chemicals. And then some things can get through the membrane, either getting inside of the protocell or outside of the protocell. And the idea is that outside of the protocell, you would have the normal chemistry. And then inside, because there is this filtering, you would have a different type of chemistry. So let's put these random protocells. So the protocells are generated randomly, which means that their membranes are all different but random. You put them in the system, and initially, not much happens. So these protocells, they are generated randomly. They don't do anything. They filter some chemicals. And so the dynamics of the system don't change much. So let's add some properties to these protocells. Let's make them first divide. So now the protocells, they absorb some material. They build up their own membranes, and when they become too big, they divide. It's already a little bit more interesting. Let's add some more complexity. So now the protocells, not only they divide, but when they divide, the repartition of chemicals in the membranes is not equal. So maybe in the 2 daughter protocells, this one will have more A, this one will have more B. So that's a type of mutation. So now we have self-replication, mutation, and also they can interact with each other. So 2 protocells, they collide, they might exchange chemical, or one might pop, or they might just absorb each other. And now we get some much more interesting dynamics. So these are all the same type of simulation, just on a larger time scale. So what you see is, for example, this purple chemical, which is to be rare in the non-protocell attractor, now can become much more common. And if you run the simulation much longer, you see that it doesn't settle into any kind of attractor state. You can run it very long, and there is nothing like a cycle or one type of stable dynamic of the system. And this is happening because when you have this mutation, self-replication, and interaction, that is enough to get the protocells to evolve. So now the protocells, initially they are random, but then some of them are better able to replicate, and so they are selected for, and you get more of these protocells. But as you get more protocells, the environment changes because these protocells, they are using chemicals, they are producing chemicals. So now the environment is not ideal anymore, and the protocells have to evolve to adapt to this new environment that they created by themselves. Here you have the total number of protocells. You can see that, for example, here, some innovation happened that made the protocells very efficient at replicating, and then they all got extinct. So they did something wrong. They invaded the whole environment, and then they all got extinct. And then some more things happened, and so the idea here is that maybe this complexity of the time series can be used as a biosignature. And so, or rather, as a signature of something is evolving in the system, evolving in the sense of mutations and selection. So we are trying to use these results in real experiments with real protocells. Sadly, this is not sulfuric acid, which sounds very exciting. Now, this idea that life and complexity go together is not my idea. It's an old idea that already exists. And the idea is that life tends to bring systems to this state of maximal complexity, which is just before chaos. And we used these ideas in a paper that we published a few years ago, completely different project. Here, the idea is that you have a planet that you're observing. You're observing it from very far away, so you only have 1 pixel. You measure the light from this 1 pixel. You measure the complexity of this time series of light. And we showed that you can relate this complexity that you measure to the complexity of the surface of the planet. So from the complexity number that you get, you can tell whether the planet only has 1 type of surface, several types of surface, so, desert and water and forest or whatever. Whether you have several surfaces plus clouds, etc. So in these 2 projects that I talked about, I use time series. But the reality is that usually we do not have time series about exoplanets. We have 1 measurement for 1 planet, if we're lucky. So can we work with that? In these different projects, that's what we try to do. So we assume that we don't have time series. We have 1 measurement per planet, but at least we have many planets. So you have a lot of data. Can you define any type of interesting biosignature? So here we define what we call a distributed biosignature, that is a pattern that is distributed on several planets, based on 3 attributes of life. So the first is life tends to spread. So we assume that it's possible in this simulated universe for life to go from 1 planet to another, but whether it is bacteria being ejected by an asteroid or humans sending rockets to other planets. The other thing that life does is life tends to change the environment. So if you have trees somewhere, they produce O2, that is a change in the environment. So we call this terraformation. And the last thing is that life is also changed by the environment. So life spreads, it mutates, and it also changes the environment. And so the question is, when you have this process of spreading and mutating and changing the environment over several planets, can you differentiate this process from other possible processes that would also produce high correlations in your measurements? And the answer is that yes, you can actually. So we did many different simulations depending on the kind of life that you are considering. And we used a measurement called the mental correlation, which is normally a measurement used for in biology, in ecology, when you're trying to make the difference between some genes that exist because of random genetic drift and some genes that were selected for. So the idea is that we can use this measure to tell us whether the patterns in the planets was created by a process that spreads and mutates or by any other type of process. So this mental correlation, it gives you a number, but more importantly, it gives you a p-value that tells you how confident you can be that this pattern was really created by a process that spreads and mutates, whether you want to call this process life or not. And so the answer is that yes, we can make the difference. This p-value goes down very, very fast. You can get high confidence pretty early in the simulation. You can define some other types of measures that help you actually localize these planets that you are looking for, the planets that are driving this mental correlation up. So with our simulations and our measures, what we find is that you can define detection process that basically gives you no false positives. So we have some planets that were terraformed here. The detection method will not detect all of the planets, but when you have a detection, you can be fairly confident that it's a real detection, that it's not a false positive. So I talked about simulations at different scales. So scale of chemistry, the scale of a galaxy. I have other simulations at other scales, but I won't talk about them because I don't think I have time. But what I want to say is that even without a definition of life, we can use simulations of artificial life to define some types of biosignatures and hopefully apply these new measures to real data. And the methodology is very simple. You make a simulation, you give the simulation some attributes of life, you find some measure that discriminate between these attributes, this simulation with and without life, and you look for processes that also are adversarial to your measure, so because you want to make sure that whatever you're measuring is really due to life and not to some other processes. I also have some simulations that simulate the emergence of entire ecosystems. And here the idea is to find whether there are some energy constraints where below a certain level of energy, you can have evolution of maybe one species, and above a certain level of energy, you may have a more complex ecosystem. And that's all from me. Thank you.